Hi everyone, it's Kristen, aka K-Dubs the Hiking Scientist on Instagram, and I have been thinking about making this video for a long time. Every semester, I teach at a local university, and I encounter students who have, one, never even touched a microscope, two, made a wet mount in order to look at samples on a microscope, or three, know that there are different types of microscopes. So it can be a little confusing while I'm teaching class if students don't have this base knowledge. So I was hoping to make this video for students that I have in the future, and then also to help anybody who wants to look at tiny cool stuff learn how to use a microscope and what type of microscope to use depending on what you're doing. Now, if some of you have followed me on Instagram, you probably see me do stuff with fungal cultures that look like this all the time. I'm a plant pathologist, so I look at a lot of fungi. You can use this video if you want to be a microbiologist, study bacteria, do all sorts of stuff. But you probably think, how am I going to get stuff like this to look at with a microscope? Well, this is just fungus. You know what else is just fungus? This. So if you're going to follow along with this video and use it as a reference, just get some hot dog buns. And I promise this is not from my house. This doesn't, like, I kept this for this video, so don't worry, I'm not living in filth. My office is, though. This is the office pot. So anyway, let's get started. So first off, there are two types of microscopes that I use on a daily basis as a plant pathologist. I use, one, a dissecting scope. So if I were to use my example of the hamburger bun, which is covered in mold, I can use this dissecting scope to kind of see a couple different things. So these dissecting scopes are kind of the first microscope that you'd use. It normally has a range of about 10x to 40x, depending on how much money you want to spend. This is about a $100, $200 dissecting scope. I turned it on with these little lights right here, and this enables me to magnify things to about 100 to 400 times, that 10x and 40x. At this point, the magnification is kind of irrelevant. We're not really measuring anything. We're just looking at things through these ocular lenses here to see if there's anything that jumps up at me. Maybe I don't know that this is bread mold. I want to make sure that I'm getting a fungus instead of getting a bacteria or nematodes or some kind of abiotic stress. I want to see if I can actually tell that there is a microorganism there for me to use down the road when I get to my compound microscope. So the first thing that I would do is look for the actual organism, and then I can also see with a dissecting scope, is that organism causing any kind of damage to my hamburger bun? But in my case, I would use something like a leaf or maybe a plant, because you can use this dissecting scope to see that if there's areas where the plant has become really watery because it's being dissolved, or maybe there's some kind of growth coming out of it. But I'm going to show you what this looks like through the dissecting scope. And fun fact, a way to remember this, dissecting scope, I can get my hands under there with tools to dissect this wonderful specimen. Alright, so this is with the dissecting scope. You can see that hamburger bun there, and I can actually see, and I'm moving the focal point here. If I go up to magnify away, see I lose my focus point. So I can gradually go up and down to find out where the best area for me to focus is. And you can also zoom in even more here with the top notch. But see how we can go through an intermediate on how to focus on this very large object? This is different from a compound scope, and we'll get to that in a few minutes. But here, if I were to be using this at work, I would say yes. That looks like a fungal pathogen of this hot dog or hamburger bun. And there we go. And I'm able to zoom on and see some actual structures that I can't see with the naked eye. So now that I've looked at this hamburger bun and seen that yes, it is fungus, it's what I'm interested in, I can now make a wet mount and sample some very tiny microscopic features about this, maybe to tell what kind of fungus it is. We can do that with a compound scope. This is a compound scope, and it is a microscope that has many different lenses. So if we look here, I can rotate these objectives. These columns here are called objectives, and they are different lenses of different strengths of magnification. So your ocular lenses are up here, it's where you look in, and they are at about 10x always. So these guys magnify things 10 times. Then these here are additional multiplications. But I'm telling you all about these numbers because this is, with a compound scope, this is where the magnification matters. Now there are two lenses here, and this is one of the things that always confuses my students. They'll see these big objects in them, and I'll show you to a second. One of them usually has a scale bar in it, and this is where the magnification comes into play, because you can know the magnification and do a math equation to figure out how long an object is that you're looking at with your glass slide here. On the other one, we'll sometimes have a pointer, and I'm going to show that to you right now. So this is through one of the eyepieces, and see that black line there? That's called a pointer. That pointer is used to identify an object. It's not part of your sample. In fact, there's actually not even anything on this microscope right now for us to look at. This is just the lens. So that pointer helps you distinguish things while you're looking with a friend maybe, because not everyone is going to know what a world reticulate kinetophore is, but you can be sure that they're going to know how to look at the end of that pointer needle to distinguish something. So that's there, and it also, let's see if I can do it, it can also move. So you can get it out of the way. You just do that by rotating the lens. Now if we look in the other one, let's see if I can do this again. This is going to be the lens that has the scale bar. So the scale bar, whew, man, this is tough. Aha, there we go. So the scale bar enables you to measure objects. And let me get a sample on there so I can show you what it looks like. So here is a sample. I put a rhizobia root nodule on there, and look, I'm able to measure it. This lens, again, that scale bar, oh sorry everybody, that scale bar is in the lens. So if I rotate this, let's just see where it ended up. Oh, look at that. It moved. Alright, so back over here with the compound scope now, you might say, let's look at our sample, I'm ready. Doesn't work. We need to make something called a wet mount. Now, a wet mount is just a slide. I'm sure you've heard about slides. It consists of a glass slide. You can see that there. You can buy these on the internet pretty dang cheap. We buy them all the time. It involves using a plastic cover slip, which can be, well, it can be plastic or it can be glass, but I use the plastic ones. And it's just a thin plastic thing that I'll show you how to put onto your glass slide then. And the reason it's called a wet mount is because you use water. Some people use stains, maybe they use something else in order to better see their microorganism, but we're just going to use water now because honestly, this green mess is going to look pretty good. Okay, now, first things first. I'm going to show you how to make a wet mount, and then we'll check out what this fungus looks like underneath this compound scope. Now, this might seem like it's pretty easy, but I have a lot of students who get kind of confused by it because they've never done it before, so it's fine. But I'm here to show you the best way to do this. So we have our glass slide on the flat surface here, and we're going to use a water dropper here to put a little bit of moisture on. And this is where you would have your stain, or maybe you would stain your sample separately, but for us, we're just going to use water. Now, this is important. Watch how much I put on the microscope slide. That's all I do. I have a lot of students who will do something like this. No, don't do that. The reason why, when you add your cover slip, after you put your sample, of course, if you were to put your cover slip on that, all that water would fly all over the place. And if you have a very tiny sample in there, it's going to blast out all over the table and not stay on your microscope slide here. So next thing we do is I have my bead of water there for my wet mount. I need to go to my sample. I have here in my hand a dissecting needle. You could use whatever you wanted, a fork. I don't know what you have at home. But all you need to do is get a little bit of this fungal stuff here. So I'm just gonna scrape a little bit. I got some of my dissecting needle here, and then just, there it is. Look at that, beautiful. And that might even be too much, so we're in for a pleasant surprise. Next, take my cover slip, and I don't just blast it on there. I don't wanna have any bubbles or anything. So I'm going to lay it down kind of at an angle, see that? And then see if there we go, perfect. So the water, that little tiny water droplet versus that mess, perfectly displaced underneath the weight of the cover slip there. And all of those fungal spores now and fungal tissue are arranged in a way that I actually have a flat surface for this microscope here to focus on. Now there's something important with this. Now so that's water underneath there, right? So if we want to look at this for, whoa, if we want to look at this for like a hot minute underneath our microscope here, which I'm going to slide the glass slide underneath here, put it on the lowest concentration objective. I'm going to put that right there. Now, this is how a microscope works. It uses light. So if I have light blasting up there for hours, my sample is probably going to dry out. What you can do, if you need to look at this for longer than 10 minutes, you can actually do something like this. You can put nail polish around it. And I made these years ago, and they're still good. I can still look at the samples because there's a little bit of a leakage there, but most of the water is still in there just because I put nail polish on and let it dry, of course, to preserve these samples. Here's the next roadblock I find with my students. They don't know how to focus a microscope. I'm going to show you a cool little fun way to do it. When we put on a sample, like we just did right there, the students will look in and they think maybe that it's in focus right away. You need to focus a microscope. We can't see anything there, and that's the point. There's no focus, it's just a blur because we haven't actually found our object. Now, a lot of students spend like 20 minutes just moving around on the microscope with this knob here, trying to move around the objective to find something. They're just scanning back and forth looking for anything as they look in these ocular lenses. Well, what you can do, if you can actually see your sample, is try and find it. Just look right at this point here instead of through the lenses and try and find what you're looking for. We can see all those fungal material there. But let's say you had something that you didn't know if it was there or not. Like maybe it's a really finite nematode or something like that. What you can do is take a Sharpie and just put a little black dot on your lens in the corner where you don't think your sample would be. And you can cruise around then. You can cruise around and look, you can find that. You can place your marker within that circular beam of light because that is what's going into your lenses for you to look at. Now, if we try and look up here, it's still going to be a blurry object, I promise you, but I can see the edges of something. So I now have something to focus on. I'll now use these over here, and they're on both sides. They're very nice for left-handed and right-handed people. But this is where you make big changes in focus and then fine changes in the focus. So if we do that, oh God, this is gonna be tough. I've never done this before through a camera. I can do something new today. Wow, done, there's our Sharpie. So I use the big knob to focus, and now I'm using the finite knob to get those edges perfect. But now that I have that focus and I know I'm at the same focal point of being just on that surface of that slide, because that's the whole point of the cover slip, right? It's making one surface for you to look at. We can now move around with these knobs and find our sample because we are on the same plane, right? We are actually on the same point. So, looks like I have something right there. Let's see if we can see a ton of spores. Yep, there they are. So we're on the same point of focus now. But they are, this is using just the lowest magnification objective. So I'm actually going to change it and increase it. And now you're able to see probably some minute detail of these structures. And every time you do this, you have to refocus with that fine focus knob. And like I was saying, every time you do this, I'm refocusing with that fine focusing knob. And those are some spores at a higher magnification. And I'm going to get it to the point where it's awesome. And I'll show you what it looks like, what your bread mold looks like, because I know you're dying to know. Are you ready? So bread mold is in the genus Penicillium. And there are all the spores. And right there, that's a structure in the middle that holds the spores called the conidiaphore. Let's see if we can zoom in on here too. Oh, double whammy. Love it. There it is. Look at how that conidiaphore, that hyphal strain, or the hyphal segments, are holding up the spores. And those little dots all around it are the tons and tons of spores of penicillium that are going to infect the rest of the bread mold that's there. Pretty cool, huh? That's not even on the highest magnification for this. So we're able to see things that are very characteristic of a genus. That structure with those spores all on top of each other like that is very distinct of this genus. And we're also able to tell minute features about the spores that can lead us to discover what kind of species this is.